Coming up ahead in this episode of Xtalks Spotlight.
Some of those clonal hematopoietic mutations can be misidentified as somatic mutations that are unique to the tumor. That matters because we might treat a patient based on a genomic aberration, thinking that that aberration is unique to the tumor, that drug is going to work well because we have evidence already.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're asking questions about clonal hematopoiesis and cancer genetics. How are they linked, and how is research evolving?
Clonal hematopoiesis, marked by the expansion of blood cells originating from a single clone due to genetic mutations, becomes more common with age and may occur before the onset of blood-related cancers. In this Xtalks Spotlight edition, we will explore how genomic profiling can distinguish clonal hematopoiesis from mutations found in tumor cells. This underscores the vital importance of germline sequencing in accurately identifying true somatic events, thereby preventing errors in diagnosis and treatment decisions. To help us shed light on this important topic, I recently sat down with Dr. Karina Eterovic, R&D Director of Oncology Diagnostics at Eurofins Viracor BioPharma Services. Dr. Eterovic is a renowned scientist and recognized leader in precision oncology and has been solely responsible for the inception, leadership, and strategic direction of a new oncology diagnostic program at Eurofins Viracor. Thank you for taking the time in this Spotlight interview, Dr. Eterovic.
Yeah, I'm happy to be here. Thank you for the invitation.
So to start us off, what exactly is clonal hematopoiesis, and why is it important in the context of cancer genetics?
Yeah, clonal hematopoiesis is not a new concept or phenomena, but it has been increasingly studied in the context of cancer genomics lately. And the definition of clonal hematopoiesis is the expansion of a single clone of blood cells, and that happens due to the acquisition of mutations, and we call those somatic mutations. As we age, acquiring mutations in general in our DNA happens to all of us, in all of our cells, but when that happens specifically in certain types of blood cells, which we call hematopoietic stem cells, that's what we call clonal hematopoiesis. With next generation sequencing, which is a fancy way to call DNA sequencing, being more and more affordable, we have been noticing that is a higher prevalence of this, I'm going to call CH mutations for simplicity, of the CH mutations more than we thought before. For the most part, those mutations in general, if they're not in blood cells, they are normal to have low impact in human physiology, but in blood cells, they can have a bigger impact that we're going to discuss later here. It's important to say that it's a phenomenon that happens with age, so there's a lower prevalence, around 1% of those mutations in patients that are 40 years old or younger, but a much higher prevalence. It could be higher than 15% in patients that are older than 70 years old. In the concept of cancer genomics in general, why we're talking about this lately, why we're studying more this phenomenon, we have seen a higher prevalence of CH mutations in patients with solid tumors. It is known already that CH mutations can increase the risk of hematological malignancies, which are not solid tumors, but we have seen several studies have shown that we can see a high prevalence in patients with solid tumors. Some of the studies have shown in about 20, 25, sometimes a little higher prevalence of those mutations in solid tumors. And why that is relevant, it's because when we study cancer genomics in general, what we do is to sequence tissue or tumor from patients or circulating tumor DNA using blood to identify molecular markers or genomic aberrations that can guide us to treat the patients. A lot of the drugs that have been developed in the last 10 years and approved by the FDA are targeted therapies that have a molecular marker associated with it. Some of those molecular markers are mutations. So, and here's where the area starts to become a little bit gray, because some of those clonal hematopoietic mutations can be misidentified as somatic mutations that are unique to the tumor. And that matters because we might treat a patient based on a genomic aberration, thinking that that aberration is unique to the tumor, that drug is going to work well because we have evidence already. But that aberration might be a clonal hematopoietic aberration. And if the patient is treated based on that, we are not going to see efficacy for that drug. So that's why it matters so much to identify and not misidentify clonal hematopoietic aberrations versus tumor-unique somatic aberrations.
And how can clonal hematopoiesis be differentiated from tumor somatic mutations in clinical practice?
Yeah, that's an important question, because the only way we can really differentiate what is a clonal hematopoiesis mutation versus what is a tumor unique somatic mutation is by sequencing in a lab both either a piece of the tumor or cell-free DNA and also germline. And by germline, I mean blood. In a clinical setting, that matters because that's not what we do for the most part. Most of the assays that we have available, if it's not under a research protocol, we will only sequence the tumor. And the problem when we do that is that everything that comes from the tumor will also reflect some of the germline data as well. The main reason why it's not in the clinical practice, I mean, the main reason, but there are several reasons, but the main reason I think it's because the ads really cost. And in the financial side, it can be complicated for approval from insurance, or if the patient is paying out of pocket. Right now, although sequencing it's a lot more affordable than it was 10, 15 years ago, it's still fairly expensive, mostly if a patient has to pay out of pocket. So when you have to sequence both tumor and germline, that will add to the cost, and that's why for the most part we don't do that. But it is the only real way to differentiate those two types of somatic mutations.
Now, in the absence of germline sequencing, what strategies can be used for inferring clonal hematopoiesis somatic events and distinguishing it from tumor somatic events?
Yeah, and in the absence of germline sequence, there are some strategies that have been used in the clinical setting. The first one is that there are common genes that are known to be involved and represent clonal hematopoiesis mutations. So there are certain databases out there that can show you the prevalence of some CH mutations in those genes. So if you sequence the tumor only without germline, and if you see a mutation in those specific genes, that is a little bit of a red flag, or at least, okay, take a step back and pay attention. This might not be unique to the tumor. This might be a CH mutation. So that's number one. Number two, it's based on the variant allele frequency. If you're not familiar with DNA sequencing, variant allele frequency refers to what is the prevalence in that DNA? What is the percentage of that mutation within that DNA? It is expected that clonal hematopoiesis mutations will show up in lower variant allele frequencies. To give you one example, any other germline mutations, those that I mentioned at the beginning that have little effect on human physiology, they will be in between 50 and 100% variant allele frequency. For CH mutations, they are in the germline, but they are in a lower range of VAF. In the third way to really try to identify what is tumor mutation versus CH is based on patient age. CH mutations are more predominant in older patients, but interestingly, we have seen in our cohort, and this is data that I'm going to give you a little spoil here, data that we're going to present at ASCO this year. This is a study that we did, and we saw the presence of some of these CH mutations. I mean, this study, we sequenced both tumor and germline, so we could confirm exactly what is unique to the tumor, what is CH. in this study, we saw some interesting CH mutations that were identified higher variant allele frequency, which is not quite expected, and also in younger patients in our cohort. When I say younger, like around 40 years old. So although there are strategies, so three strategies, one based on the gene that is involved, second, variant allele frequency, and third, patient age. Some of the studies have shown that still cannot assure you that and make you differentiate what is CH and what is a mutation that belongs to the tumor. It's going to be very nice to present that data at ASCO later this year. It's going to be in June.
Now let's talk about hematologic malignancies. What role does clonal hematopoiesis play in the risk of developing these cancers?
Yeah, we know that there is a higher risk of developing a hematological malignancy for when patients present, or when people in general present CH mutations. Additionally to that, which is very relevant, it's CH mutations can also originate from cytotoxic therapy treatment, like chemotherapy or radiation. And the problem with that is that some patients, for example, that might have recovered from a solid tumor but went through cytotoxic therapy, those patients can be at risk of developing a new hematological malignancy in the future, and that can be really tragic. So the risk in general is still low, but we need to keep monitoring because there is a risk of developing hematological malignancies with CH.
And to bring us back to our main question, how is research evolving in the field of clonal hematopoiesis, and what future directions do you foresee?
Yeah, we have more and more studies now that we can go back when you have tumor data, in data means sequencing, when you have sequencing data from the tumor and we have germline, people are going back to that data, investigators are going back to that data and try to see and learn more from that data. That's the same thing that we did here with a prospective study. So the way I see this evolving is obviously with more sequencing. We need to learn more. We need to learn more about how those mutations, clonal hematopoiesis mutations, are really created. I mean, there are pathways there that we don't know yet. What are other factors that might be involved other than age or treatment with cytotoxic therapy? What is really happening there, and what can be done? And I think, of course, not much can be done even when we identify a patient with a higher, or a person with a higher level of clonal hematopoiesis mutation, but we need to keep monitoring. And we need to keep monitoring mostly those patients that might have been treated for other types of tumors as well. But I do see the field evolving. I do see a little bit more effort coming to maybe make it possible in a clinical context to sequence both germline and tumor. Again, there's a financial component there. It's not only the financial component, sometimes it's hard to get both samples at the same time. And but I think we need to really pay attention and try to do that as much as possible and find ways to make sequencing even cheaper, and find ways to put in the clinic tests that can do both at the same time without being a financial burden for the patient.
Well, thank you very much, Dr. Eterovic, for speaking with us today. We really appreciate your time and insights.
Thank you for having me.
We look forward to learning more about Eurofins Viracor BioPharma Services work in oncology diagnostics. Thank you all for joining us for this Xtalks Spotlight feature. We hope you enjoyed the discussion.